Let me start this slideshow. Okay, so today we are going to talk a little bit about a particular type of BMI, and I had posted a paper which I'll also put in the YouTube box below so that people can look at it, and some work that's been done in this field. But this is a specific device that a group is working on. It's a hippocampal prosthetic device. We're going to talk about what they are, and I'll introduce myself. I'm Regina Monaco. I am the founder of this company, CDSS, which is the Center for Dynamical Systems Studies, and we work to study dynamical systems, of which the brain is one. So this introductory picture I'm going to describe because we're going to talk a little bit more about this. So just orienting you all to what this is going to help understand this. So the hippocampus, which I will say more about, so don't worry if you're not exactly sure what it is or where it is, but it's a structure in the brain, deep in the brain, in the temporal lobe. I'll say more about that in a few slides, but this is a picture of it, and it's intimately, in fact, essential for the encoding of memories. If you don't have a working hippocampus, you don't encode memories. You might have some short-term recall of where the bathroom is, or that this is a cup of coffee, but you won't remember something you've seen and that's new in 5 minutes. So the hippocampus is essential for making that kind of encoding those kind of memories in your brain. So this picture is from the paper that I posted and that we'll be discussing. Anyway, this would be the input from short-term memory, the kind of thing that you just see immediately and remember for a few seconds. And then this is an artificial device that's implanted in the surface of the brain, but it's an invasive device, and it will have electrodes that go through the brain into this biological region called the hippocampus. And the input goes through this device, goes through these electrodes, and is re-encoded by this device into the hippocampus. And that enhances the ability of the memory to work. So if you have some issues with memory, this prosthetic device will enhance it. So I will say much more about that in a second. So today we're going to discuss this specific paper done by researchers at the University of Southern California and the Wakefield Medical Center in North Carolina. I think it's important to note that this is heavily funded by DARPA, and you will see in a few slides, but I'll say now, this research started in the 1990s, and DARPA has funded many projects that are all along these lines. So it's good to remember that they're cutting edge. They fund cutting edge research, but they also are part of the United States military. Go ahead.
OpenBCI was their first project was funded by DARPA. That's how it started.
They're very intimately involved in this research, and it's good for taxpayers to understand what we're funding. And so the particular point was to make a brain-machine interface to enhance memory encoding. These are invasive devices. In the first talk, I defined that there are non-invasive, semi-invasive, and invasive devices. Non-invasive would be the EEGs, which is why we can put this together as a group, because like a hat you put on. Semi-invasive sit on the surface of the brain, but you're still going in there, right? It's still a surgical procedure. And invasive is electrodes penetrating into the brain at various depths. There's a variety of invasive devices. This is one of them. In fact, this is one of the deepest invasive devices because of where the hippocampus is. So it uses very long electrodes, much longer than Neurotech that you would use. Those are millimeters deep, and they go into the cortex. This is going through the brain tissue into the hippocampus, and I'll show you in a slide where that's located. So deep into the hippocampus, into very specific subregions called nuclei, and it will both read and write information to the hippocampus. So if you remember, I said a minute ago, if you remember with your short-term memory, that I said how intimately the hippocampus is involved in memory encoding, then if you're reading and writing to it, you're affecting / interfering with memory encoding. And interfering can be good, right? If you have trouble encoding something, interfering could also be enhancing. So, and I didn't say this enough in the first meeting. I might say this every time. When we talk about these things, it might seem that the hardware design is the most important part of a BMI, right? It's a physical thing. You can see it. People are putting it together. I think it's really important to remember that it's the discovery and understanding of how to decode the natural neural codes. That is I wrote here, equally difficult technical challenge. It might be more difficult. I guess it would depend on who you ask, but it's an incredibly challenging problem. If you think about how the neurons are positioned in the brain and how so many things are not only projecting to so many regions of the brain in a correlative fashion, but also how many things are happening dynamically and at the same time. And then you have to understand how that's happening. And then you, the researcher, have to replicate that in an accurate way, or else you're just going to send nonsense into the brain. The technical challenge is incredible. And so I think you always have to remember that the modeling and the neural encoding, understanding the neural encoding, I think is really the heart of these. I think the hardware is not that hard. So now this is all to show you what we're talking about with the hippocampus. This is not specifically about the device. So this is a picture of a brain, and I also have a little toy brain here. It's not a great toy brain. In fact, my cat chewed it up a little bit, but it gives you an idea, and it matches these pictures a little bit. So this right now is facing as if a person is looking this way. This would be the back of the brain, and this would be the front. And this lobe, which is not really that well defined, but you see a little bump here, that's the temporal lobe. The hippocampus is in this lobe. And in this picture on the left, you can see these structures, which I'll say more about in 1 second. But you see this walnut colored thing. It's not really here, but that's the cerebellum. And that would be actually right under here, under the occipital region. So I know I'm throwing a lot of terminology if you're not familiar with the brain, but you'll hear it over and over again. These are very standard areas, and it just gives you the idea that the brain is one thing, but it has all these regions, and each region has a different job. So the hippocampus, now you have a little better idea. If the temporal lobe is here and it has some thickness, the hippocampus is in the middle of it, and people will say it's in the medial temporal lobe. So that's all that means, is that it's in the side here, and deep inside your head, like maybe 2 inches inside, like over your ear, an inch up, and then 2 inches in, and that would be where your hippocampus is. And you have one on each side, and they sort of have projections that go towards the back, the rear of your brain, towards I just dropped my brain, towards your occipital region. So this shape, see, this is your hippocampus. This is the basal ganglia. It's also part of encoding memory, but we're not really going to talk about it today. I'm just saying it because it's a very common area to speak about. So it does feed information into the hippocampus. And the cerebellum I just talked about, and it also affects the memory encoding. So there's a lot of stuff going on, right? There's neurons that are projecting into the hippocampus. And this is if you rotate the brain and then cut it down the center. So now the front part of the brain is behind the page. The occipital region is behind us here. And this is now, so now you can clearly see that there are 2 hippocampi, and here's the cerebellum sort of behind it. Oh, you know what? I think it's the other way. I actually think that it's facing this way, but it's still a slice through the middle because the cerebellum would always be in the back, and I see there's a brainstem here. So this should give you some 3-dimensional idea of hippocampus. Now you know what we're talking about. Here's another picture of the hippocampus. So we're really going to fill in what this means. So here's the brain, right? I orient you guys. Here's the cerebellum. Here's the hippocampus. Now this one is in pink. And this is a little better picture. It's got this long process coming out of it, and it's sort of wrapped around the thalamus, which is another region that you'll hear a lot about. So now you have a really good idea of how deep inside the brain the thalamus is. So when you hear, not to do with this talk, but people doing deep brain stimulation, right? They're going in, they're opening the skull here, they're putting a device on the surface of the brain, and then they're threading electrodes through the cortex, right through whatever this blue region is, through the corpus callosum, through the amygdala, into the thalamus. That is a lot of structures that it's going through, and a lot of tissue that it's piercing to get to the thalamus. So, and the thalamus is right next to the hippocampus. So you get an idea of how deep these structures are. They're really deep within the brain. So the hippocampus, and now I can't read my own slide because it's covered by the pictures of the attendees. But see what I remember. The hippocampus basically takes input from a variety of sensory areas via highly interconnected subregions. Oh, and I want to go back and say this. So again, I said this in the first slide, there's a lot of different types of neurons. They look very different, very morphologically distinct. There's a type of neuron called a pyramidal neuron, and it's got a lot of fuzzy dendrites on one end and a lot of fuzzy dendrites on the other, which means it can make a lot of dense interconnections. And if anybody here does machine learning or AI, so you know about neural networks, computational neural networks, and they have all these hidden layers. And the more hidden layers you have, the more parameterization you can do, and the larger the computational matrix that you can manage through the system. So if you use that understanding of neural networks in engineering, you can see that a pyramidal cell with this a huge amount of dendritic spikes coming out on each end and axons on the other, it's equivalent to a large neural network with a lot of hidden layers, and that's why it can do so much computation in the brain. So I think that that's a really important fact to, I keep saying remember in this talk about hippocampuses. But anyway, so it is composed of these highly densely connected pyramidal cells, which give a very dense array of communication. And as I already said, it's in the medial temporal lobe. Now we understand what that means. So there's a specific pathway that the information comes in. So I want to say that the basic idea you should come away from is that information comes in through your sensors. So I'm showing you fingers for the sense of touch, your eyes, right? Visual, through the nose, through the, name all 5 senses, right? They all come in through physical interaction with your environment, and they come in through neurons that are physically in these organs, in your eyes and your nose and your fingers. They go in through the spine, so they come up through the spinal column, and they come up into the cortex, right? They kind of distribute throughout the brain, but they all project back to the through the thalamus to the hippocampus. So I think the overview is that you get physical information from the environment, and your brain then encodes it into memories after it's processed in the brain. And I think that that's a great simple model to come away from. So I'm going to name subregions. I promise you I'm going to show them in a picture in a second. So there's a subregion of the hippocampus. These are all parts of the hippocampus. Data comes in through the entorhinal cortex. It passes through something called the dentite gyrus. It passes through 2 areas, the CA1, C3, CA1, and I can't see the last piece, but I think it ends at the subiculum. And then there's some projecting back to the cortex and back to another part of the hippocampus itself, and that's all for like feed forward and feed backward information for double checking. The brain actually does a lot of that double projection throughout, like cortical columns and other structures in the brain. So this encodes long-term memories from short-term memories. So, and to the way that the hippocampus, you can't remember everything, right? Obviously the brain, everybody's brain, like everybody can go to a circus or a movie and they're going to remember different things. They all were in the same room. They all got the same sensory data. So there's obviously other things than just the raw input of information that's determining what the hippocampal encodes. And so it can be determined by your emotion, where your attention and focus is, if it's a repeated action or if it's brand new, and what you assign as an importance to it. If you're bored and you're not paying attention, literally you won't remember something that maybe the person next to you will remember very sharply. So just because you're in the room and getting sensory data doesn't mean you're encoding it. Your brain is also actively filtering what you're going to remember. And that's done in this whole limbic system area, the thalamus and the hippocampus, the hypothalamus, which is not in this picture, the amygdala, they all work together to determine what the hippocampus will encode. So,
is it okay to ask a question?
I'm sorry?
I have a working mic now. Is it okay to ask a question?
Yeah.
I don't want to interrupt.
No, that's okay.
I somehow never appreciated, actually, if you can go back one slide, showing the left and right. Is that like really, I'm looking at the right image, that like the memory is really stored in such distant locations for left and right, or is that sort of just not to scale? Because that implies that, I'm assuming it's just, the sensory stuff from the right ends up getting into the left, hemisphere. So it's like our memories of like what happened on my left really being stored separately from what happened on my right.
Yeah, that's actually, I didn't cover that here, but there's been some really famous experiments done. I don't know if you know about the corpus callosum, which is a thick, dense, communication pathway between the two halves of the brain. And they've done studies. Some people had to have the corpus callosum cut, they have epilepsy or whatever, but then you can study what happens when this communication pathway is cut. And you have to read more about it, but they literally behave as if they have two brains. And they've done experiments where they sort of block like what the left eye and the right eye sees. Yeah. And the brain
you familiar with the
cannot
Kim
integrate.
Peek?
Go ahead. I didn't hear
with
you.
Kim Peek?
Kim Peek?
You know, the Kim Peek, the inspiration for Rain Man.
You know? Oh, I didn't know that was who that was.
He was born with no corpus callosum.
I didn't know that.
He's like a super savant.
That I knew from
very
the movie.
Strange. But no corpus callosum.
So usually the whole point of the corpus callosum is so that the two halves do integrate. But if that pathway is broken, the brain does act a lot like there are two brains in your head. So
but is there like storage? I'm sorry to interrupt. I'm sorry.
Oh, that's all right. Is there stories?
Is there like storage in the corpus callosum? Because I can imagine it. I'm picturing the hippocampal region as there being storage there, of memories.
Yeah.
And in the corpus callosum is sort of like a cable, maybe allowing communication.
Yeah.
But if there's no like storage, then it's not really like an extended part of the hippocampus, and so they really are sort of separate, but allowed to communicate.
Yeah.
So that still feels like much more separated than I appreciated before with the left and right hemispheres.
Yeah. Well, it's like a disease to have the corpus callosum not broken. It's part of the system. It's supposed to be there in a way, so they don't ever, like when they're acting separately, like in those situations, it's very abnormal. Like they can do it, but it's sort of a broken system. So these are separate organs, but they're supposed to be highly interconnected through the corpus callosum, to always be communicating with each other. So
I'll make this my last question for now. Like if something happens with my left hand and with my right hand, I kind of have like it was with my hands type association. So, but maybe in the short term, at least, these are being stored, the sensory stuff is being stored separately, but is there some sort of like migration to like, there's maybe my dominant hand, that hippocampus side gets like, ultimately is where that whole type of memory goes, and my other, so that like still it ends up being like, hands are in one region, and not like memory of left hand, memory of right hand, separate histories.
So I think I'm understanding your question. I think I want to break the idea that there's a location of where memory is. Memories are stored diffusely throughout the brain. And then there's memory could be thought of as the neural circuitry, the correlated neural circuitry that brings all these different areas together. Wherever that's processed, probably the prefrontal cortex, but either it's not known or I don't know, I'm not sure. But, that's all pulled together when the memory needs to be retrieved. But it's pretty clear that there's a very diffuse encoding and no one single location of where a memory is stored. And in fact, part of the reason for that is for robustness of recall, because if part of the brain is damaged by aging or by, a knock on the head or something, you can lose parts of memories, but your memory, your recall will feel like you've got the whole memory. It might be harder to access it, but because of the redundant and diffuse encoding, you'll be able to pull back the whole memory. It might take a little longer as people get older. They might say, oh, give me a second, it'll come to me. And that's because parts of the network are weakened or maybe gone, but eventually it all comes together. And that's because of that diffuse encoding throughout the brain. So, Okay, sure. So I think we just discussed this. So we got to my wonderful picture. I couldn't find this anywhere on the web, so I had to draw it. And this was sort of fun. But anyway, so this is my artistic rendition of a hippocampus, and see this thicker end. Now I'm just going to go back to orient you guys. That's this. So my picture is sort of going the other way. So this is that body that I'm going to talk about in a second. And then this would be, you're going to hear me say subiculum and entorhinal cortex and dentate gyrus. And that's all along this what I'll call a tail. So, and so if you read anything in the literature and you see these words, now you'll know that it's really very simple. So this is the hippocampus, right? And it's got these regions. Here's the dentate gyrus. And then these subregions called CA1, CA2, CA3, CA4, they go around counterclockwise. Then you go a little further up the tail, you have the subiculum, you go a little further out the entorhinal cortex, and you won't hear we won't say anything about this today, but the tip of the tail is the parahippocampal gyrus. And information, and then there's a structure called the fornix, which is feeds into the hippocampus, but is actually a separate structure with a separate name. And then information comes in from the thalamus to the fornix, and then the fornix into the hippocampus. But the not official, right? But the input generally comes in from something called the perforant pathway. It's just upstream, right? We're only talking about the hippocampus now. And it goes in this picture now, you get the input from let's say the cortex, right? The perforant pathway is getting it from your cortex. Your cortex is getting it from your motor pathway and from your sensory organs. So remember I said that earlier. So it goes in, here's in the tail, the entorhinal cortex, and goes in means that's the connection of those neurons, right? So the entorhinal cortex, and then that has projections, jumps all the way to the dentate gyrus. That's just how those neurons grow. If you're going to ask me why isn't this next to this, or why is the subiculum in the middle, ask evolution. Like I don't really know why, and no one knows why. It's not a question of why. This is the design. So it goes from entorhinal cortex to the dentate gyrus. Then it goes, and I numbered it. So it goes 1, 2, 3 is the dentate gyrus. Then that projects, its neurons talk directly like a phone line to the CA3 region. And every step of the way, there's processing. The neural circuitry is doing something. It's not just moving it around. There's processing of the data. CA3 then projects to CA1. And then it ends up at the subiculum, which is here. And the subiculum does two things. Output means it goes back to the cortex where some of it, where it came from, right? Motor neurons go to the cortex feeds it into this. It goes back to the cortex where then your conscious mind becomes aware of whatever memory you're trying to recall. And it also projects backwards to the entorhinal cortex, and which then projects out to the prefrontal cortex. And while this is very convoluted, if you study brain networks a little more, you'll see that there's always this kind of looping where it goes through the system and then it loops back on itself. There's structures called cortical columns, which maybe we'll talk about at a future meeting if people are interested. And they have several layers. And the layers, which you could call these layers too, right? They're just areas that have different functions. And the cortical columns have internal projections that are very similar, right? So they process the data and then they pass it to another part of the cortical column, which does the same processing, like in a redundant fashion. And then they project outward to whatever the next step in the data processing is before you do whatever the thing is, recall something, learn something, act on something. So the brain is highly interconnected and hierarchical in that way. And I can't see this. You guys, the picture of all of the attendees is on top of this, but I see what I was trying to say. So this is another picture. I'm just naming the lobes of the brain, right? The occipital lobe I can't see, but I know that it's back here. The parietal lobe is here, right? So right where my bun is right now, the frontal lobe is here. The temporal lobes, like we said earlier, on the side. Sometimes when I talk about the brain, I'll make a little hand puppet, and the temporal lobe is your thumb. And that's actually a pretty good model, if you're just casually talking about the brain. And then the occipital region would be the end where your wrist is, and the frontal would be here, and the parietal would be on the top. So in this picture, it's just another way of me trying to orient you. There's a structure called the basal ganglia, which I might have mentioned this earlier. I don't remember. But in any case, it does have something to do with feeding some input into the hippocampus, but here's the hippocampus sort of nestled into the medial temporal lobe. So these are I'm just trying to orient you guys. All I've done so far is say, what's the hippocampus? We haven't started talking about the paper, but you'll be happy to know that there's only a couple of slides on that. So now that we know what the hippocampus does, and it encodes memories, and if you think about it, there's kind of nothing more important that a mammalian brain in a conscious creature can do than encode memories, right? That's how you learn about your environment. That's how you learn about yourself. That's how you are social. That's how you have relationships. Like it's really the most important part of having a brain. If you learn something, I mean, I think we saw the movie with Dory the fish, right? She remembers for like a minute and then she forgets. It was a kid's movie. And it was cute and everything. But the point is, you can't live an effective life if you don't have a memory. So I personally think there's nothing more important that your brain can do than have good memory. So if you start losing your memory, that's everybody's fear as they start getting older and facing the fact of aging, right? Not everyone loses memory, but there's enough people that do that people are afraid of it. So enhancement of it is it seems like an important goal. Every time I talk about BMIs, I always try to make people aware of the possible nefarious use if they're not controlled right or they fall into the wrong hands. So I could also say that a device put into your brain that can enhance your memory can also ablate your memory. So just be careful what you wish for. And as these are developed, I think every scientist in the field has to make sure that they have security on them, that they're under complete control of the user, with every BMI possible. But that being said, let's put it aside now, and I'll say the good things. So first, who can have bad memory? I already alluded to this: people with dementia, people with Alzheimer's. Then things that you might not think of. There's an enormous amount of memory loss with injured professional athletes. They end their career in their 30s or 40s maybe, and they almost universally, especially in football, have a lot of impairment. And so, poor memory is very bad, and they're already young, not already young, very young. So they have a lot of life ahead of them. Injured athletes would like to not have that kind of damage to their brain. Just normal aging, health has been increasing until COVID happened. We had what's called an aging population, at least in the Western world. People were routinely living to late 80s, 90s, even 100. But there's a natural memory loss. And, if you're going to live older and want to be healthy, having a memory enhancement can help you live a more comfortable, more productive life. So there's a lot of good reasons to have a memory prosthesis. Then you could also really want enhancement if you have no medical issues, right? Just because you're a student and you want to learn faster or more or easier. Athletes just to learn whatever. I don't play any games, but I know there's a lot of complicated things in planning in football or whatever. And I guess you have to remember the steps in order. And so a good memory would help you do that, like being a dancer or something. Actors, things that have a lot of demands on your attention all the time. Pilots, surgeons. I was just making things up, fields that people need memory. Professors, right? If you're learning something all the time, you're all anybody who wants to learn and is spending a lifetime enjoying learning or has to learn for their job is going to benefit from accepting a memory prosthetic device to do purposeful memory enhancement. Now, remember that memory is not always great. Anyone who has eidetic memory, which is like, total recall all the time, acts like it's a curse. They say they hate it. They can't separate the memory of like what was on page 32 of the last book they read, and they can read it back to you, but they can't separate that from a birthday party or a more meaningful memory. They don't have the ability to sort the encodings by importance. And every person that's been interviewed that truly has eidetic memory has said that it was a burden. So you don't want too much memory, and you definitely want it under control of the limbic system, where you pick and choose what you're remembering. People with PTSD or trauma are going to have memories that they don't want. So maybe having the ability to erase memories, which, by the way, has already been done in the literature with this device, could be helpful for them. But then you have to remember, if the device can erase memories, you have to be careful with how it's used. So we'll just keep in mind that there are some memories that people don't want to write or encode, and would like to erase or just weaken in their brains, in their minds. So now we get to the point, the paper that I posted, and I don't know if people had a chance to look through or read, but the hippocampal prosthesis, which is a BMI that's definitely implanted physically in the hippocampus. So this project began, actually I wrote here around 2005. It began in 1998, and it was designed to understand, I wrote the specific problem of memory encoding. It's deeper than that. It was designed to understand how the brain goes from taking sensory input to what kind of firing patterns and what kind of pathways does that data go through in order to turn from an experience into a memory, into a long-term memory, right? Which is different from a short-term memory, as we established earlier. And then I wrote here what I just said, that it's under the direction of the hippocampus, but it's informed by these structures. And now you have an idea of what they are, right? The thalamus, we saw that earlier, the fornix, which is a little tail on top of the hippocampus, and the cortex, which is the outer cells on the brain, which cover all the deeper structures. That's all your cortex. And all areas of the cortex, especially the prefrontal cortex, which is most highly associated with attention and complex tasks, and the formation between giving your attention to complex tasks and the formation of memories. So the hardware was designed by this group, Dong Song and Hampton and Ted Berger, all the authors of that paper. Oh, there's one other guy, got a terrible name, like Wade Killer or something. It's on the next slide. But this group got together in the early 2000s and made this device. And the device exists. It's actually already patented. And I'll just say this, there's different kinds of memories, but we're talking about declarative memories as opposed to the memory of riding a bike, like the memories have to do with skill. And then people will talk about episodic memories. That just means I remember an episode in my life, personal life experiences. And semantic would be like me talking about different parts of the brain. If you come away from this talk and you say, I know the structures, I know where the subiculum is and the hippocampus, that's more of a factual memory, or it's a little different from a life experience. And the other kind of memories are procedural. So now we know that there are different kinds of memories in addition to short-term and long-term memories, and they're encoded differently. And so this group took it upon themselves to understand the different encoding between all these different kinds of and types of memories that are stored in the brain. So the first successful implant, this might be the last slide, by the way, yes. So the first successful implantation of this device was in 2019. And I looked all over the web for a picture of just the device. So last week we talked a lot about Neuralink. There's tons of pictures of Neuralink. You can see the device. It's a coin-shaped device. It's the thickness of your skull. It's got electrodes coming off of it. There's a lot of photos of it. I couldn't find any picture of this except what I showed you on the first slide, and I'll show you something after this as well. But Deadweiler is the name. I couldn't remember. It has the word dead in it. It's sort of a weird name. I don't want to say creepy, but there it is. I just said creepy. I'm sure he's a very nice guy. So these three guys plus Ted Berger, who I left out, wrote this paper, which you can look for. It's separate from the talk that I posted. And here's a quote from the paper, which I thought encapsulated the whole thing. They developed a hippocampal neural prosthetic to facilitate human memory encoding. I can't read it. Your pictures are on top of it. To use the patient's own spatiotemporal neural encoding. What that means is how you naturally fire, what your brain does as you encode a memory, right? You see a picture, you read a book, right? They're measuring, they've got these electrodes already in your brain for whatever reason, right? You're an epileptic and you agreed to have them implant them, or you know, whatever you've gotten into their hands, you've agreed to have this implanted. So the first step was understanding how the encoding happens. That means you give the patient a task and you read out the neural encoding from electrodes that are placed in all those different regions of the hippocampus. Remember, I said there was a subiculum and there was a dentate gyrus, a CA1, CA3, CA4, and CA2. And so they had different electrodes in each region, which, by the way, is not that easy to put in properly. So there's a lot of work in just figuring out how to do that. I could give a whole nother 30 minutes on that. But in any case, they had the electrodes there. They get the data out. Then they have to make sense of it. There's an enormous amount of correlative functions that are used to study this sort of nonlinear dynamical system. And that's why I say I think this is the hardest part. I don't have a single slide on this because the level of the math and the interpretation is very high. We can do a future talk on it if people want, but I really think this is the most challenging part of I mean, getting an electrode into somebody's brain and just reading the outputted data. You know, I don't know. I feel like we could do that if it wasn't illegal, you know, on a table in a back alley. But this part of understanding the data, that's something. That's an enormous mathematical engineering achievement. So anyway, so they were reading the spatiotemporal neural data and then determining how its firing patterns lead to encoding of information. And then they replicate those firing patterns and see if they actually do replicate the process of the formation of a memory in the hippocampus. So, and then this says that they developed a nonlinear multi-input, multi-output model. That means it has an array of electrodes, multi-input, multi-output, right? Multiple electrodes will do that. And what they were studying is the regions, remember, in the bulk of the hippocampus, the head part had that CA1, CA2, CA3, CA4 regions, and that the dentate gyrus fed into the CA3 and then fed out through the CA1. That's what they primarily studied, was the encoding between those two areas and the kind of spike train firing that was happening as you encoded information. And that's exactly what this says, the patterns of CA1 neuron firing during the encoding phase of a human memory task. So they did that. They found some neural codes. They wrote a paper on using this MIMO model that they devised. They put a device into somebody's head in 2019, and they were astonished. They were looking for maybe a 5 or 7% increase. You know, that's normally what you'd find with an initial trial, because they knew that they didn't really know what they were doing, and models don't always work. They enhanced memory and recall of specific learning tasks by 35%, which is astounding. And this is the first model, right? This is 4 years later now. I can't imagine what they've done, because it's not all published. I'm sure that it's better by now. So, and then I just wanted to point out that the team began working around 1999. And then again, with this particular guy, I think he must have been a graduate student, and I don't know whose lab he was in. And then he joined the Song and Deadweiler lab, and then he got back to this work in 2010. So there was a bit of a gap in here. There was an article in Wired magazine in 2004 on exactly this topic. So they had to have some kind of early working model. I couldn't get the article because it's behind a paywall, but it's worth looking up, especially if you have a subscription to Wired. And I'm going to try to get a copy of that article. And then I'm listing here what the work was, which I kind of just described, right? The first thing you'd want to do is localize and test the electrodes that are put into the various areas, right? The CA1, the subiculum, the dentate gyrus, and you want to look at the data that's coming out. You have to interpret it, right? What comes out is not just like a single clean line. It's going to have noise. It's going to have artifacts. It's going to have crosstalk. So the first decade was spent on just getting clean data and separating the data from these different areas that are very physically very close to one another. Then you can start to and look at the encoding patterns that you want to copy. And then from there, it's sort of a straight shot. Once you've figured out the correlative patterns between how the data is encoded through the hippocampus, then you can start to copy it and replicate it and then re-upload it to the brain through the electrodes. That part is much easier, actually. So the decoding, this is what I just said, and development of a non-linear stimulatory input model. That's another way of saying what I just said. And that happened as early as 2015. Then they did a variety of experiments in animals. Then they built the device. Then they got the FDA approval. Then they did the successful implantation in 2019. So they found that the only successful working model is this multi-input, multi-output device, which I'll show you in a second. Is the only picture I could find was in the talk that I posted. And this device received a US patent in 2017, which is 2 years before they went to the hospital and tried it on patients. So I'm going to stop sharing this to share the next piece. I have to close this. So I don't know if you guys, did you guys get a chance to see the Dong Sung talk that I had posted on the Meetup site?
I looked at it today, actually. It was
okay.
Yeah.
Yeah.
Yeah. Okay. So then go ahead. So I was thinking of maybe showing a couple of minutes of it, but I don't think that's necessary. Let me close this though. Oh, I don't remember making changes. So let me just minimize this. I don't know what I did. I'll clean this up later. So, oh, I thought I had it open in a YouTube. Let's see. What I'm trying to do is pull up the talk because there's like a critical 2 minutes where I think he gives the bulk of the essence of the talk. Here it is. So I'm going to share this again. Okay, so right around like 4 minutes and 15 seconds, you see this picture. I don't know how you guys feel. I thought this talk was almost impossible to understand. Everything's working against you. The guy has a very strong accent. The interpreter, if you turn on closed captions, it gets half the technical words wrong. So you have to actually kind of know what he's saying to read the captions, because when it makes mistakes, you're like, oh no, he said hippocampal and not, I don't know, to nickel campus or something. So, but now that I've given this talk, if you go back to this, I don't know if you guys really want me to play this or not, but if you just go back to this part, you don't have to listen to the whole thing. Maybe I'll just talk about this for a second without playing it. So he gives the only picture I've seen of the device, right? So here's the MIMO model, and the multi-input, multi-output means he's got a bunch of electrodes. So that's the multi-electrode array. Those would project from this device, which I'm assuming is just a chip with it does some local compute functions and then collects the data from the electrodes. And then the electrodes are physically, attached to the chip. This is how these things work. And I'm sorry. I saw at the top Dong Sung. I don't know if you guys see this. And I thought for a second he joined the talk and my heart just fell out. It's like, oh, is he here? I'm talking about his work. So, so the output is the electrodes that project into the hippocampus. And then if you listen to just the 2 minutes, right, between 4 and 6 minutes, he says exactly what he does. Right? He calls it a biomimetic device. It directly interfaces with the hippocampus through these electrodes, and he's transforming what he calls the upstream hippocampal signals. What I believe he means is the input of the short-term memory goes through this, is processed locally on this chip, and is output into the hippocampus. So it bypasses any damaged hippocampal regions that maybe aren't doing their job. And instead of looking for input from the hippocampal region that might not be working because it's been damaged, say by Alzheimer's or something, it provides that output directly into the hippocampus, which allows the encoding of long-term memories. It's really well done. It's a beautiful design, as long as you don't mind having, a BMI device implanted deep into your brain. And I think if you have a disease, like my mother-in-law has Alzheimer's, and I I think, I mean, you can't ask her, right? Because her memory is completely gone. But if someone said to me, you're going to be like this, you have Alzheimer's and it started, and in 3 years you're going to be, do you want to be like this, or do you want this device in your head? I would rather have the device in my head. Because when you lose your memory, you lose yourself. So that being said, I'm ready to take questions or hear your comments. And thank you very much.